Hey everybody, welcome to our final section of this chapter on nephron transporters. So now we're going to talk about the collecting duct. So the collecting duct is the final, it's the home stretch for us in the kidney, right? We've filtered our substances, we've gone through the PCT, the loop of Henle, the distal convoluted tubule, and now we're in the final area of the kidney, of the nephron, where we can alter the concentration of our urine, we can alter the pH, and really make an effort to get all of the substances we want from the body, and then excrete the rest of the ones that we don't need or we don't want at this time. So if you remember our little nephron that we've been drawing, we've had the PCT highlighted in yellow, the loop of Henle highlighted in green, the DCT in light blue, and now we're in this kind of collecting duct portion, which I'm highlighting in orange. So right off the bat, we need to be asking ourselves, what is the goal of this portion of the nephron, right? Each portion of the nephron had individual goals, but ultimately we're working together kind of as a team to get the maximum amount of substances that we need out of the urine and then excreting the things that we no longer need or no longer want for the body. So the collecting duct really has two primary roles, which are in conjunction with some of the other portions of the nephron and very similar. So one, it's to reabsorb sodium, water, and urea. Remember, our primary goal is to reabsorb sodium because that water will then follow. The principle of osmosis says that water will go from a low solute concentration to a high solute concentration, and thus if we reabsorb sodium into the body, this water is essentially going to follow it and tag along. The same is true for urea, which we haven't talked about much, but the same concept exists. Urea is a solute. So if our body reabsorbs urea, the water is essentially going to follow because it wants to go to the area that has more solute concentration. This is good for our body. We don't want to be getting rid of our water most of the time. We want to keep our fluid levels, our volume status at a good, healthy level. And if we're just continuously getting rid of our water, this could be problematic. Just because it's been a while, a quick refresher here. Remember, when I say we reabsorb a substance, that means it goes from in the nephron back into the body, versus if we secrete a substance, it's going from the body, the interstitial space, the bloodstream, into the nephron to ultimately be excreted from the body as urine. Next, I want to draw your attention to this diagram that we have, which talks about the different types of cells in the collecting duct. So when we think about the different types of cells in the collecting duct, we want to think about what is their primary function. And if we think about what their primary function is, then it makes sense in terms of what kinds of transporters we would expect to be in that location. So first, let's look at the principal cell, which is this cell right here that I'm highlighting in yellow. There's a lot going on in this cell, as we can see. This is definitely one of our workhorse cells in the collecting duct. But let's break it down and look at the transporters that we might already be familiar with and then say, okay, well, which ones are new? Which ones do we need to add to our knowledge base for understanding this portion of the nephron? So right off the bat, you're probably recognizing our classic sodium-potassium ATPase, 3 sodium going out, 2 potassium going in. But on the apical side, the side that is actually facing the nephron, look at all these cool transporters that we have, including one that will reabsorb sodium. And ultimately, because we have the sodium being reabsorbed, we've said that water is going to follow, right? Water is going to follow sodium because it wants to go from low concentration to a high concentration of solute. So my question for you, why is it that sodium moves into the cell here with this transporter? And the answer is to remember that if given the opportunity, solutes want to move down their concentration gradient. If sodium is being pumped out the sodium-potassium transporter on the basolateral membrane, that is going to decrease the amount of sodium inside the cell, and thus it provides a lower concentration relative to how much is out in the nephron tubule. So if given the opportunity, solutes are going to move down their concentration gradient into the cell. Remember, this is the example when we talked about the crowded bus, right? If you're crowded, cramped in with a bunch of people on the bus, as soon as those bus doors open, you're going to get off as soon as you can to get more space from the crowdedness around you. So then you could probably make sense of my next question, which is why would potassium move into the nephron if there was a transporter, if given the opportunity? And the same principle applies. Potassium is actively being pushed into the cell. That creates a large amount inside the cell. So if given the opportunity to get off of the crowded bus, get out of the crowded cell, it's going to do so and move down its concentration gradient. So those are transporters you're probably familiar with. Notice, though, there are 2 receptors that you're probably less familiar with, and that is this V2 receptor, which is coupled with ADH, as well as this receptor, which is coupled with aldosterone. We'll talk about that shortly when we talk about hormonal effects that can alter the collecting duct and the reabsorption of certain molecules. The next important cell type in the kidney is this alpha intercalated cell. I'm going to talk about this in conjunction with the beta intercalated cell below because these have very similar properties in that they are kind of like the opposite. Remember when we talked about the loop of Henle, there was a balance between the thin descending limb and the thick ascending limb, but despite them having several key differences, they were ultimately working together in the physiologic process. So if we contrast the alpha versus beta, notice that one secretes acid, the other reabsorbs acid, and vice versa is also true. in that the alpha reabsorbs bicarb and the beta secretes bicarb. So the key for me here was just to put the alpha sign with acid and the beta sign with bicarb, but you have to remember that that's talking about secretion, and then it essentially has the opposite effect on the other molecule. So if I just remember that the alpha intercalated cell secretes acid, and I know the beta intercalated cells are the opposite, then I can reason from that if alpha secretes acid, it must reabsorb bicarb, and then the beta does the opposite. It secretes bicarb and reabsorbs acid. Notice these alpha intercalated cells also carry one of these interesting aldosterone receptors we're about to talk about. And the other key point I want to highlight here is the difference between these transporters which use ATP and these transporters which do not use ATP. So typically a general principle is that we use ATP when we are pushing things against their concentration gradient. Again, this should make sense, right? If we're bringing potassium into the cell, we know that's against its concentration gradient because there's a lot of potassium in the cell, and that is why we need this ATP for an active transport process. We need energy to create this push to get molecules through the cell. When we're pushing them, we're stuffing them into the cell, which is against their concentration gradient. So now let's step back and talk about these very important hormonal effects on the collecting duct. So the two important ones to know are aldosterone and ADH, which stands for antidiuretic hormone. So aldosterone is a very important mineralocorticoid that our body secretes as part of the renin-angiotensin-aldosterone system. We have a whole chapter on that system, so if you need a refresher or you're interested in more of a deep dive, then go ahead and check that out. But the key here is to remember that aldosterone does a lot. It has a lot of actions in the collecting duct, and the primary goal of aldosterone is to reabsorb sodium so that we can then reabsorb water, because aldosterone is primarily secreted when our body senses we are at a low volume status. So that it acts to then secrete aldosterone to then bring back sodium, ultimately bringing back more water and more volume. So as you can see here, aldosterone binds to a mineralocorticoid receptor, which then goes on to alter messenger RNA and ultimately protein synthesis. So let's look at some of these effects of aldosterone in the cell. I've highlighted aldosterone in pink, and that's the color that I will use to show which areas of the cell, which transporters are actively affected by aldosterone. So first thing is aldosterone is going to stimulate the sodium-potassium pump. Why? Because again, we want to bring in more volume status. In order to do that, we need to bring in our sodium so that the water will follow. It's also going to affect the channel on the apical side, which is this sodium channel. This is called the ENaC. ENaC stands for the epithelial sodium channel. This is also an important channel because we can target with pharmacotherapies, which we will talk much more about in the pharmacology section. A key point here is that by stimulating this ENaC channel, we're creating something called lumen negativity in the nephron. And what that means is, because we're bringing across a positively charged ion in sodium, we are actually pushing out other positively charged ions, such as potassium. So stimulation of these ENaC channels and bringing back sodium indirectly pushes potassium out of the cell as well. One, because if you bring more positive charges into the cell with a bunch of sodium, that is going to repel another positive charged molecule such as potassium. Remember, we talked about the magnets. Two positives will repel each other. And two, creating a negative environment in the lumen of the nephron is actually going to pull that potassium across. So now we're in a scenario where potassium in the cell is both being repulsed by an electrochemical force, and it can move down its concentration gradient by leaving the cell. And thus, if given the opportunity, it's going to do so. And so this is how aldosterone can actually increase potassium secretion. So notice the one thing we haven't talked about yet is that it also increases hydrogen proton secretion. So not only does aldosterone affect transporters in the principal cells, it also affects the hydrogen proton ATPase in the alpha intercalated cells. So by stimulating this ATPase right here, you can see that that's actually going to push more and more hydrogen protons out of the cell into the nephron to ultimately be excreted. So the key here is that aldosterone affects many different transporters within the collecting duct, ultimately working to reabsorb sodium and water at the expense of potassium and hydrogen ions. And the other hormone we're going to talk about here is ADH. So ADH binds to the V2 receptor with the ultimate goal of reabsorbing free water. So if we draw our attention to the V2 receptor and the ADH molecule over here, the binding of ADH to the V2 receptor will actually cause the release of vesicles right here that then go to the other side and add additional water transporters in the nephron. These are called aquaporin channels. So aquaporin channels are also called water channels, and essentially what they do is they make it easier for water to pass through the cell. Our body is going to reabsorb and hold on to that water much more easily than if we didn't have these transporters or these channels available. And the final thing that we're going to talk about to wrap up this video is, again, our renal tubular defects. So we have two here. We have one called Little syndrome and one called syndrome of apparent mineralocorticoid excess. Sometimes this is abbreviated SAME. So Little syndrome is a gain-of-function mutation that decreases sodium channel degradation in the collecting duct. Because of this, we have more sodium channels, an inability to degrade them, and thus we reabsorb more sodium over time. If we reabsorb more sodium, we've talked about how this is going to affect our volume status as well. This presentation is very similar to something called primary hyperaldosteronism, because the primary effect of aldosterone, we said, was to reabsorb that sodium. And what are the effects that can also occur if we have a bunch of aldosterone? They're very similar to the physical exam findings we would see in a patient with Little syndrome. So we get a metabolic alkalosis. Why is that? Because remember, one of the effects of aldosterone was that it stimulated this potassium ATPase, and thus we're secreting a bunch of our proton ions, ultimately to be excreted. This is going to create an alkalotic state. It's going to raise our blood pH. Hypertension as well should make sense to you, because if we're bringing back sodium and we're bringing back more water than we actually need, right, because this is a defect in the transporters, that's going to raise our blood pressure. If we increase our volume status higher than what it should be, that's going to have more volume in our vascular system and put more pressure on the walls, ultimately leading to hypertension. And finally, hypokalemia, or low potassium in the serum, because again, if we bring back a lot of sodium molecules, this is going to create a scenario in which more potassium is actually secreted into the tubule and ultimately excreted in the urine. We talked about that being due for two reasons. One, an electrical reason, and one, a concentration reason. And then here it is. It's finally here, the autosomal dominant. I told you that a lot of these renal tubular defects are autosomal recessive, but there's one that's different, that is autosomal dominant, and that is Little syndrome. So again, I think a good strategy here is to remember that Little syndrome is autosomal dominant, and then you can just remember that the rest of them are autosomal recessive if it were to show up on a test. And the final one we'll look at is syndrome of apparent mineralocorticoid excess. Again, the name tells you a lot about what's going on here, and we really don't need to get in the weeds as far as the biochemistry in terms of how this process works, but you do need to understand that the ultimate finding is essentially a state of low aldosterone. So ultimately, this is a loss of function mutation where we lose an enzyme that then causes us to have an increased amount of cortisol in the body. So normally this enzyme would convert cortisol into cortisone, but if we have this genetic condition where our enzyme is essentially wiped out, we are unable to proceed with this reaction, and thus we're going to get a buildup of cortisol. And you might be saying, why is this a problem? Why does this cause a presentation of apparent mineralocorticoid excess despite the fact that we have low aldosterone? And the reason is because cortisol can actually stimulate the mineralocorticoid receptors and produce a patient presentation that is similar to those that have high levels of aldosterone, despite the fact that they actually do not. So put another way, high levels of cortisol will then stimulate the mineralocorticoid receptor, which we said was the receptor that aldosterone normally stimulates. And so it's going to create a presentation that is very similar to hyperaldosteronism. And you can see this by looking at the physical exam and the presentation. There are similar findings to that of hyperaldosteronism or Little syndrome. So again, you'll get a metabolic alkalosis, hypertension, and hypokalemia or low potassium. We talked about why that is with Little syndrome. So think about the name here. It's syndrome of apparent mineralocorticoid excess, because we don't really have an excess amount of aldosterone. That's why in the lab we'll actually see low levels of aldosterone. But because we have so much cortisol, which are then stimulating the receptors, we are getting the same ultimate effects in the end. And the final thing that's worth knowing is the genetic inheritance, which is autosomal recessive. All right, so that's it. So that's our nephron transporter series. We talked about the proximal convoluted tubule, the loop of Henle, distal collecting duct, and now the collecting duct. Feel free to watch these over and over again if you need to really understand the concepts. We're going to continue to build on these nephron transporters, particularly in our chapter about the renin-angiotensin-aldosterone system. So great job, everybody, and I will see you in the next one.